Hello, and welcome to Calm Science Nights. I'm so glad you're here with me tonight. Before we begin our descent into the deep ocean, if you find peace in drifting off to the soothing rhythm of scientific discovery, please consider subscribing to the channel. It helps us create more of these tranquil explorations together. Now, settle into your most comfortable position. Let your body sink into the mattress like a stone settling gently to the ocean floor. Feel the weight of the blankets around you like water pressure, comforting and secure. Allow your breathing to slow and deepen, like the gentle rhythm of waves far above. Tonight, we're going to descend into the last great unexplored frontier on Earth, the deep ocean. A realm of darkness and mystery that covers more than half our planet, yet remains less mapped than the surface of Mars. Over the next 100 facts, we'll trace humanity's journey from the sunlit surface down into the crushing depths, discovering the remarkable science of the abyss, told as a story of exploration, wonder, and revelation. There's no need to stay awake for all of it. In fact, the greatest compliment you could give is to let sleep take you whenever it wishes, perhaps somewhere between the twilight zone and the midnight zone, drifting into dreams as we drift into darkness. Just allow these words to wash over you like a gentle current, carrying you peacefully into rest. And so the foundation was laid, the Challenger's revelation that the ocean was far deeper than mountains or tall, that life thrived in the crushing darkness, that the ocean was layered and slowly circulating, that food drifted down like snow or arrived in rare windfalls of whale carcasses, and that pressure increased relentlessly with every meter of descent. These early discoveries opened the door to the abyss, but the journey had only just begun. So many mysteries remained in the darkness below, waiting for those brave or curious enough to descend. Feel yourself sinking now, like marine snow drifting gently downward through darkening waters. There's no urgency, no struggle, just a peaceful descent into comfort and rest. Let the story carry you down. As the 20th century dawned and technology began to advance, the dream of actually descending into the deep ocean started to seem achievable. Engineers and scientists turned their minds to the problem of pressure, designing vessels that could protect human observers from the crushing forces of the abyss. The breakthrough came in the 1930s with a man named William Beebe and his engineer partner, Otis Barton. Together, they designed what they called a bathysphere, a hollow steel sphere just 4.5 feet in diameter with walls 1.5 inches thick enough to withstand the pressure, yet small enough to be manageable. The sphere had 3 small windows made of fused quartz, the only material they could find that wouldn't shatter or deform under pressure. Inside this cramped metal ball, barely large enough for 2 men to squeeze into, Beebe and Barton would make history. The bathysphere couldn't move on its own, it was simply lowered on a steel cable from a ship at the surface, dangling like a weighted pendulum into the depths. In 1934, off the coast of Bermuda, Beebe and Barton descended to a depth of 923 meters, just over 3,000 feet, deeper than any human had ever ventured. Through those small quartz windows, Beebe became the first person to observe the deep ocean in its natural state, and what he saw captivated the world. Beebe's descriptions of the descent, radioed to the surface as he descended, were both scientific and poetic. He described how the colors disappeared with depth, first the reds and oranges, absorbed by the water within the first 10 to 20 meters, then the yellows and greens, until only blue remained, a deep, penetrating blue that gradually darkened toward black. By 200 meters, The water around him was a dark twilight. By 300 meters, the darkness was broken only by occasional sparks and flashes, bioluminescence, the living light produced by deep-sea organisms. The deeper he descended, the more frequent these lights became, like stars appearing in a darkening evening sky. BB counted hundreds of lights in a single descent, brief flashes from jellyfish, glowing lures dangling from the heads of anglerfish, trails of luminescent mucus left by swimming creatures. He described fish with enormous eyes, adapted to capture every available photon in this near darkness, and other fish with eyes so small they seemed almost vestigial, species that had given up on vision altogether in the perpetual night. The bioluminescence that BB witnessed so abundantly was one of the deep ocean's most remarkable adaptations, though understanding it would take decades more research. Scientists eventually discovered that approximately 90% of all deep-sea animals produce light through bioluminescence, making it perhaps the most common adaptation in the largest habitat on Earth. The light is produced through a chemical reaction involving a light-emitting molecule called luciferin and an enzyme called luciferase. When luciferin is oxidized in the presence of luciferase, it releases energy in the form of light, typically blue or blue-green light, because these wavelengths travel farthest through seawater. Different species use bioluminescence for different purposes, some as lures to attract prey, like the famous anglerfish with its glowing fishing rod, some as camouflage, producing light on their undersides to match the faint light filtering from above, erasing their shadow when viewed from below, some as communication signals to find mates or coordinate with members of their own species, and some as defense mechanisms, releasing clouds of glowing chemicals to startle or blind predators in the darkness. While BB's bathysphere opened the first window into the deep, it had severe limitations. It could only descend where the cable allowed, and the cable itself was a danger, as its weight increased with depth until it risked snapping. What oceanographers needed was a vessel that could move independently, exploring the ocean floor freely rather than hanging helplessly from above. This dream was realized in 1960 when Swiss engineer Jacques Picard and U.S. Navy Lieutenant Don Walsh descended to the deepest point in the world's oceans, the Challenger Deep in the Mariana Trench, in a vessel called the Trieste. The Trieste was a bathyscaphe, a fundamentally different design from the bathysphere. It consisted of a small pressure sphere for the crew, similar to BB's design, but attached to a large float filled with gasoline. Gasoline, being lighter than water and relatively incompressible, provided buoyancy without being crushed by pressure. To descend, the Trieste carried iron pellets as ballast. To ascend, it released the ballast and floated back to the surface. On January 23, 1960, Picard and Walsh reached a depth of 10,911 meters, nearly 7 miles down, the deepest place on Earth. The descent to the Challenger Deep took nearly 5 hours, sinking at a rate of about 1 meter per second through increasing pressure and darkness. At these extreme depths, the pressure reached approximately 1,100 atmospheres, enough to compress the Trieste's steel sphere by nearly a millimeter, enough that Picard and Walsh could hear the metal creaking and groaning around them as it deformed under the load. When they finally reached the bottom and turned on their external lights to look out through the small viewport, they saw something that shocked the scientific community: life. Even here, at the absolute deepest point of the ocean, where the pressure was crushing and the cold was absolute and the darkness had reigned undisturbed for eons, They observed what appeared to be a flatfish and several shrimp-like amphipods moving across the sediment. The significance was profound. There was apparently no depth in the ocean, no matter how extreme, where life could not find a way to exist. The ocean floor was not the barren wasteland early scientists had imagined, but rather a continuum of life from surface to abyss. The discovery of life at the greatest depths raised fascinating questions about how organisms could survive such pressure. For many years, scientists assumed that deep-sea organisms must have special adaptations to withstand the crushing force. But research eventually revealed a surprising truth. For most deep-sea organisms, the pressure isn't actually crushing at all. The key insight is that most deep-sea creatures are made mostly of water, and water is nearly incompressible. If the pressure is equal inside and outside the organism's body, and it is, because water penetrates throughout their tissues, then there's no crushing force. The organism doesn't feel the pressure any more than you feel atmospheric pressure at sea level. The challenge isn't the pressure itself, but rather how pressure affects the chemistry and physics of biological molecules. At high pressure, the structure of proteins can change, membranes can become less fluid, and normal biochemical processes can be disrupted. Deep-sea organisms have evolved special versions of enzymes and structural proteins that function properly under high pressure, what researchers call piezophilic, or pressure-loving, adaptations. Some deep-sea microbes not only tolerate high pressure but actually require it to survive. When brought to surface pressure, their cellular processes fail and they die. They're as adapted to the deep as we are to the surface, living in different worlds on the same planet. The Trias's historic dive to the Challenger Deep proved what was possible, but it also revealed the immense difficulty and expense of deep-sea exploration. For decades after 1960, no one returned to the deepest depths. The ocean floor remained largely unexplored, known only from indirect evidence, sound waves bouncing off the bottom, sediment cores pulled up in tubes, the occasional photograph taken by cameras lowered on cables. But in the 1970s, a new technology began to revolutionize ocean exploration: the development of remotely operated vehicles, or ROVs, and later, autonomous underwater vehicles, or AUVs. These robotic explorers could be sent to the deep ocean without risking human lives, equipped with cameras, lights, sampling tools, and sensors. They could stay down for hours or even days, exploring areas too dangerous or remote for crewed submersibles. The deployment of ROVs and AUVs opened the deep ocean to systematic exploration in unprecedented ways. Scientists could now survey long transects of the ocean floor, creating detailed maps and collecting samples from specific locations. One of the most important early ROV missions occurred in 1977, when the submersible Alvin, a crewed research submarine capable of reaching depths of about 4,500 meters, was exploring the Galapagos Rift, an area where tectonic plates were spreading apart on the ocean floor. The scientists aboard Alvin were searching for hydrothermal vents, places where seawater seeps into the oceanic crust, gets heated by magma below, and then erupts back out through the seafloor, carrying dissolved minerals. Theoretical models predicted these vents should exist, but no one had ever seen one. When Alvin's lights finally illuminated the first vent field, what the crew saw was so unexpected, so utterly unlike anything predicted, that it fundamentally changed our understanding not just of the ocean, but of life itself. And so we've descended deeper, following the pioneers who dared to enter the abyss, Beebe in his cramped steel sphere, watching colors disappear and bioluminescent lights appear like stars, Picard and Walsh in the Trieste, creaking under pressure in the deepest trench, finding life where none was expected, the robotic explorers that followed, tireless and patient, mapping the unknown. We've learned that pressure doesn't crush from within, that organisms are adapted to their depths as perfectly as we are to ours, and that the ocean holds surprises beyond imagining, waiting in the darkness for those who seek them. Feel the pressure of your blankets around you now, not crushing but comforting, like the equal pressure inside and outside a deep-sea creature, perfectly balanced, perfectly safe. Let yourself sink deeper into rest, deeper into sleep, following the story down into the abyss. When Alvin's crew illuminated that first hydrothermal vent field in 1977, they found themselves looking at something that seemed impossible, a thriving oasis of life in the deep ocean darkness, far richer and more abundant than anything found on the surrounding abyssal plains. The vents themselves were spectacular, towers and chimneys of mineral deposits rising from the seafloor, belching superheated water, some as hot as 350 to 400 degrees Celsius, that billowed upward like dark smoke. These became known as black smokers because of the way the mineral-rich water looked as it erupted into the cold ocean, the dissolved minerals precipitating instantly into dark particles that created the smoke-like appearance. But it was what surrounded these vents that truly astonished the scientists. Clustered around the hot water flows were massive communities of organisms unlike anything seen before. Giant tube worms, called Riftia pachyptila, grew in dense clusters, some reaching heights of 2 to 3 meters, nearly 8 feet tall. These tube worms had bright red plumes protruding from white tubes, and they grew remarkably fast for deep-sea organisms, achieving their full height in just a few years. Enormous clams, some as large as dinner plates, carpeted the rocks. Strange white crabs scuttled across the vent surfaces. Mats of white bacteria covered the rocks in thick, ghostly blankets. The biomass, the total weight of living organisms, in these vent communities was estimated to be 10,000 to 100,000 times greater than on the surrounding deep-sea floor. It was as if the researchers had stumbled upon an underwater rainforest in the middle of a desert. The mystery was immediate and profound. How could so much life exist here, thousands of meters below the surface, far beyond the reach of sunlight and photosynthesis? What were these organisms eating? The answer, when it emerged from subsequent research, was revolutionary. The base of the food chain at hydrothermal vents wasn't photosynthesis but chemosynthesis, the production of organic matter using chemical energy rather than light energy. The superheated vent water was loaded with hydrogen sulfide, a chemical compound that's toxic to most life but that certain bacteria can use as an energy source. These chemosynthetic bacteria oxidize hydrogen sulfide and use the released energy to convert carbon dioxide into organic compounds, just as plants use sunlight to convert carbon dioxide into sugars. These bacteria form the foundation of the entire ecosystem, either living freely in the water or, even more remarkably, living inside the bodies of other organisms in symbiotic relationships. The giant tube worms, scientists discovered, have no mouth, no digestive system, no way to eat in any conventional sense. Instead, their bodies contain a specialized organ called a trophosome, which is packed with billions of chemosynthetic bacteria. The tube worm's red plume absorbs hydrogen sulfide, oxygen, and carbon dioxide from the surrounding water and delivers these chemicals to the bacteria living inside its trophosome. The bacteria use these raw materials to produce organic compounds through chemosynthesis, and the tube worm absorbs these nutrients directly from the bacteria. It's a partnership so intimate and essential that neither organism can survive without the other. The tube worm provides the bacteria with a safe home and a steady supply of the chemicals they need, and the bacteria provide the tube worm with all of its nutrition. This discovery meant that the tube worm is essentially a living vessel, a mobile container allowing bacteria to harvest chemical energy from hydrothermal vents. The discovery of chemosynthetic ecosystems at hydrothermal vents fundamentally changed biology's understanding of the requirements for life. For the entirety of known history, every ecosystem on Earth had been thought to depend ultimately on the sun, photosynthetic organisms capturing solar energy, and all other life feeding on them directly or indirectly. But here, in the absolute darkness of the deep ocean floor, was an ecosystem powered entirely by Earth's internal heat and chemistry, completely independent of the sun. This had profound implications not just for understanding Earth's oceans but for the search for life elsewhere in the solar system. If life could thrive in Earth's deep ocean using chemical energy from hydrothermal vents, perhaps similar life could exist in the subsurface oceans of Jupiter's moon Europa, or Saturn's moon Enceladus, or anywhere else where water and chemical energy coexist, regardless of distance from the sun. Following the initial discovery at the Galapagos Rift, scientists found hydrothermal vents in ocean basins around the world, along the mid-Atlantic ridge, in the Pacific, near Antarctica, anywhere tectonic plates were spreading apart or volcanic activity heated the oceanic crust. Each vent field had its own unique community of organisms, many of them found nowhere else on Earth. Some vents were dominated by tube worms, others by clams or mussels, others by shrimp that crowded so densely around the hot water that they seemed to form a living mat. The variety was astonishing, but all these ecosystems shared the same fundamental energy source, chemical energy from Earth's interior, harvested by bacteria and shared throughout the community. The study of vent organisms also revealed remarkable adaptations to extreme conditions. The vent shrimp, for example, were found to have specialized organs on their backs that some researchers believe can detect the faint glow of infrared radiation from the superheated vent water, essentially seeing heat itself, allowing the shrimp to navigate toward the warm, chemical-rich water while avoiding areas hot enough to cook them. Many vent organisms have special proteins and enzymes that remain stable and functional at temperatures that would denature the proteins in most other organisms. Some vent bacteria can survive and even thrive in water at 120 degrees Celsius or higher, temperatures that would instantly kill almost any other life form on Earth. These organisms, called thermophiles and hyperthermophiles, push the known boundaries of life's temperature tolerance. But hydrothermal vents presented another puzzle. They're not permanent features. A vent might flow vigorously for years or decades, but eventually the geological conditions change, the underwater volcanic activity shifts, the cracks in the seafloor seal up, the hot water flow diminishes and stops. When a vent dies, the ecosystem that depended on it dies too. Yet somehow, when a new vent opens up miles or even hundreds of miles away, it becomes colonized by vent organisms within months to years. How did these organisms find the new vents across the vast, dark expanse of the ocean floor? Research into vent ecology revealed a fascinating life cycle. Many vent organisms release larvae into the water, tiny, drifting young that are carried by deep ocean currents. These larvae can survive for weeks or even months in the water column, drifting potentially hundreds of kilometers from their parent vent. When they encounter the chemical signature of a new vent, the distinct smell, so to speak, of hydrogen sulfide and other vent chemicals in the water, they settle and begin to grow. It's a strategy similar to wind-dispersed seeds on land, but operating in slow motion in the deep ocean's sluggish currents. The deep ocean, it turned out, had its own version of island biogeography, with hydrothermal vents as isolated islands of habitat separated by vast distances of inhospitable seafloor. The discovery of hydrothermal vents and their ecosystems also had implications for understanding the origin of life on Earth. Some researchers proposed that life might have first emerged not in warm shallow pools illuminated by sunlight, as Darwin speculated, but in the darkness near hydrothermal vents. The vents provided energy in the form of chemical reactions, minerals and catalytic surfaces that could facilitate complex chemistry, and protection from harmful ultraviolet radiation and asteroid impacts that would have bombarded Earth's surface in its early history. The theory of a hydrothermal vent origin for life became increasingly compelling as scientists discovered that some of the most ancient lineages of life, based on genetic analysis, were thermophilic microbes that thrive in hot, chemically rich environments similar to modern vents. These ancient microbes, whose ancestors might date back 3.5 billion years or more, might still be living in environments very much like those where life first began. And so the story transformed, from a deep ocean dependent entirely on scraps falling from above to a deep ocean with its own engines of life, powered by the planet's internal heat rather than the distant sun. We learned of black smokers billowing their dark chemical clouds, of tube worms with no mouths sustained by bacteria within, of shrimp seeing in heat, of larvae drifting for months through darkness to find new vents miles away. We discovered that life's possibilities extend far beyond what the surface world suggests, that energy can be harvested from chemistry as well as light, that perhaps life itself began in the darkness and heat of the deep. Feel the warmth of your own body now, your own internal heat, like a miniature vent in the darkness of your room. Let that warmth comfort you as you drift deeper, following the story into stranger territories, into realms where all the rules seem different, where life finds ways that surface dwellers never imagined. As exploration of the deep ocean continued through the 1980s and 1990s, scientists began to realize that hydrothermal vents were just one type of extraordinary environment on the ocean floor. Another discovery, equally remarkable in its own way, emerged from areas where methane and other hydrocarbons seep up through the seafloor, places called cold seeps. Unlike the dramatic black smokers with their superheated water, cold seeps are quiet, subtle features where chemicals emerge from the sediment at the same frigid temperature as the surrounding ocean water. Yet despite the lack of heat, despite the seeming absence of energy, these cold seeps also support thriving ecosystems. At cold seeps, scientists discovered dense communities of organisms clustered around areas where methane bubbles up through the sediment. The methane, which would normally rise through the water column and eventually escape to the atmosphere, becomes a food source for specialized bacteria that can oxidize methane, consuming it and using the released energy for chemosynthesis, much like the hydrogen sulfide oxidizing bacteria at hydrothermal vents. These methane-eating bacteria form thick mats on the seafloor, sometimes several centimeters deep, creating carpets of white or orange microbial growth. Large clams and mussels, some species growing to extraordinary sizes with shells 30 centimeters or more across, harbor these methane-oxidizing bacteria in their gills in symbiotic relationships similar to the tube worms at hydrothermal vents. The clams essentially farm the bacteria, pumping methane-rich water through their gills and absorbing the organic compounds the bacteria produce. One of the most bizarre organisms found at cold seeps is the ice worm, or Hesiochica methanicola, which shouldn't be confused with ice worms that live in glaciers. These are different creatures adapted to a very different kind of ice. At some cold seeps, the combination of cold temperature, high pressure, and abundant methane creates a substance called methane hydrate, or methane ice, a crystalline structure where water molecules form a cage around methane molecules. Methane hydrate appears as white or translucent chunks that look remarkably like ordinary ice, but if you brought a piece to the surface and released the pressure, the methane would bubble out vigorously and the ice would disintegrate. These ice worms live directly on and within the methane hydrate, somehow tolerating a substance that would be toxic to most organisms. Scientists discovered that the ice worms are likely feeding on bacteria that colonize the hydrate surface, bacteria that in turn are consuming the methane. It's an ecosystem built on frozen gas, another example of life finding possibilities in the most unlikely places. The discovery of methane hydrates had implications far beyond their role as habitat for ice worms. Scientists began to realize that vast amounts of methane are locked in hydrate deposits in ocean sediments around the world, particularly along continental margins where organic-rich sediments and the right pressure-temperature conditions create ideal environments for hydrate formation. Current estimates suggest that the total amount of carbon stored in methane hydrates worldwide may equal or exceed all other fossil fuel reserves combined, an enormous reservoir of potential energy, but also a potential climate concern. If ocean temperatures rise or pressure conditions change, these hydrates could destabilize, releasing massive amounts of methane, a potent greenhouse gas, into the atmosphere. Some researchers have proposed that sudden releases of methane from destabilizing hydrates may have contributed to rapid climate warming events in Earth's past, though this remains a subject of ongoing research and debate. As scientists continued mapping and exploring the deep ocean floor, they began to appreciate not just the strange oases of hydrothermal vents and cold seeps, but the vast abyssal plains that lie between these hotspots of activity. These plains, stretching for thousands of kilometers in every direction, cover more of Earth's surface than any other habitat, approximately 50 to 60% of the planet's total surface area. At first glance, these abyssal plains seem monotonous, flat or gently rolling expanses of fine sediment, mostly clay, and the microscopic skeletons of plankton that have drifted down from above over millions of years. The sediment accumulates extraordinarily slowly, typically just 1 to 2 centimeters per thousand years, so slowly that a sediment course spanning a few meters might represent millions of years of deposition. It's one of the quietest, most stable environments imaginable, cold, dark, with almost no variation in temperature, pressure, or conditions over vast stretches of time and space. Yet even these seemingly barren plains harbor life, though in sparse densities. Sampling expeditions dragging nets across abyssal plains captured brittle stars, sea cucumbers, tiny crustaceans, polychaete worms, and many other organisms adapted to life in perpetual scarcity. Photographs of the abyssal seafloor often show tracks and trails crisscrossing the sediment, evidence of organisms moving across the bottom, feeding on bacteria and organic particles in the mud. These tracks provided evidence that the abyssal plains, while not as dramatically active as the surface ocean or as rich as hydrothermal vents, maintain a slow-motion ecosystem where organisms move, feed, and live out their lives on timescales that seem glacial compared to surface habitats. Some deep-sea organisms show remarkably slow metabolisms and growth rates. A sea urchin might live for decades or even a century, growing so slowly that annual growth rings in their skeletal plates are barely detectable. The study of abyssal plains also revealed something unexpected about deep-sea biodiversity. When researchers analyzed the species collected from abyssal sediments, they found remarkably high diversity, not in terms of abundance, since each square meter might contain only a few individuals of most species, but in terms of the number of different species present. A single sampling expedition might collect hundreds of distinct species of small organisms like amphipods, isopods, and polychaete worms, many of them new to science. This pattern became known as the deep-sea diversity paradox. How can such a seemingly uniform, food-poor environment support so many different species? Various theories have been proposed that the immense area of abyssal plains allows for enormous total populations even if local densities are low, that the stability of the environment over geological time has allowed species to accumulate without the extinction events that periodically reset diversity in more variable environments, or that subtle variations in sediment chemistry, grain size, or food availability create a mosaic of microhabitats that appears uniform to human eyes but provides distinct niches for specialized species. Among the most intriguing inhabitants of the abyssal plains are the xenophyophores, single-celled organisms that can grow to remarkable sizes, some species forming delicate, branching structures 10 centimeters or more across, making them among the largest single-celled organisms known. Xenophyophores construct intricate shells, or tests, from particles they collect from the sediment, tiny mineral grains, sponge spicules, fragments of shells, cementing these materials together with an organic adhesive to create beautiful, complex structures. Despite their size and complexity, each xenophyophore is a single cell with multiple nuclei distributed throughout its body. Their ecological role is still being studied, but they appear to be important in processing sediment and may provide habitat for other small organisms that shelter among their delicate branches. They're remarkably fragile. Bringing a xenophyophore to the surface intact is nearly impossible because the structures collapse without the support of the surrounding water, making them difficult to study but adding to their mystique as deep-sea curiosities. The exploration of abyssal plains has also revealed the impact of human activity in even the most remote parts of the ocean. In the late 20th and early 21st centuries, photographs and video surveys of the abyssal seafloor began to show an increasing presence of human debris, plastic bags, bottles, fishing gear, even items as mundane as a shoe or a chair. Researchers were startled to find trash in the Mariana Trench, at depths approaching 11,000 meters, about as far from human habitation as it's possible to get on Earth. The presence of plastic in particular became deeply concerning because plastic persists in the environment for decades or centuries, breaking down into smaller and smaller particles but never fully biodegrading in the cold, dark, low-oxygen conditions of the deep sea. Studies found that deep-sea organisms, from tiny amphipods to larger fish, had ingested microplastics, demonstrating that human-generated pollution had penetrated even to the deepest parts of the ocean, far beyond the reach of sunlight but not beyond the reach of human impact. And so our exploration continued across the vast, quiet abyssal plains, those enormous expanses where methane seeps create islands of ice inhabited by strange worms, where sediment accumulates 1 centimeter per 1,000 years, where brittle stars and sea cucumbers move in slow motion across mud that spans millions of years of history. We learned of xenophyophores building delicate mansions from borrowed grains of sand, single cells grown impossibly large in the stillness of the deep. And we learned that nowhere is beyond human reach, that our debris has fallen even to the greatest depths, a sobering reminder that the abyss, for all its remoteness, is not separate from us but connected to the surface world we inhabit. Feel yourself settling now like sediment drifting down through still water, gentle and unhurried, layer upon layer, building peace like the ocean floor builds depth. There's no rush, nowhere to go but down into rest, into the quiet darkness where time moves differently, where sleep comes as naturally as sediment settling on the abyssal plain. As our journey through the deep ocean continues, we turn our attention to one of the most remarkable and mysterious features of ocean life, the largest daily migration on Earth, a phenomenon so vast and so regular that it influences the entire ocean ecosystem, yet it occurs largely out of human sight in the darkness between the surface and the deep. Every evening, as the sun sets and twilight fades to darkness, countless billions of organisms begin to rise from the depths toward the surface waters. Every morning, as dawn approaches, they descend again into the darkness below. This is called the diel vertical migration, and it involves more biomass than any other animal migration on the planet. The organisms participating in this migration span a remarkable range of sizes and types. Tiny zooplankton smaller than a grain of rice, jellyfish trailing delicate tentacles, shrimp-like krill swimming in coordinated swarms, small fish with silvery scales and large eyes, squid pulsing through the water with rhythmic jets. Scientists estimate that billions of tons of animal biomass participate in this nightly movement, rising hundreds of meters from the mesopelagic zone. The twilight zone between approximately 200 and 1,000 meters depth, up toward the food-rich surface waters under cover of darkness. The reason for this enormous migration is fundamentally about survival and feeding. During daylight hours, the surface waters are dangerous for these small organisms. Larger predators can see them easily in the well-lit water, making them vulnerable to being eaten. But the surface is also where food is most abundant, phytoplankton blooming in the sunlight, creating the base of the ocean's food web. The diel vertical migration represents a compromise. Descend into the dim safety of the mesopelagic zone during the day to hide from visual predators, then rise to the surface at night when darkness provides cover, allowing safe feeding in the productive surface waters. As dawn approaches and light begins to penetrate the water again, the migrants descend, some traveling 500 to 800 meters in just a few hours, retreating to the safety of the depths. This daily rhythm has been occurring for millions of years, synchronized to the ancient cycle of day and night, a living tide that flows vertically through the ocean rather than horizontally across it. The discovery of diel vertical migration came partly from an unexpected source: sonar. During World War II, naval sonar operators detected a mysterious layer in the ocean that reflected sound waves, appearing on their instruments as if it were the seafloor, but at the wrong depth. Even more puzzling, this phantom bottom moved up and down, rising toward the surface at night and sinking during the day. Scientists eventually realized this wasn't the seafloor at all, but a dense layer of organisms whose bodies reflected sonar signals. They called it the deep scattering layer, and it provided the first evidence of the vast scale of vertical migration. Modern sonar and acoustic surveys have revealed that deep scattering layers exist throughout the world's oceans, varying in depth and density but consistently showing this daily vertical movement. Some researchers estimate that the deep scattering layer contains more fish biomass than all the world's commercial fisheries combined, a vast reservoir of marine life living in the twilight zone, mostly beyond the reach of human exploitation. The organisms participating in deep vertical migration also play a crucial role in what oceanographers call the biological carbon pump, the process by which carbon is transported from the surface ocean to the deep. When migrating organisms feed on phytoplankton and other food at the surface during the night, they consume organic carbon. When they descend during the day, they respire, releasing carbon dioxide at depth rather than at the surface. They also produce fecal pellets that sink even deeper, carrying carbon down into the abyssal zone. Some migrating organisms die during their descent or are eaten by deep-dwelling predators, and their bodies sink, taking carbon with them. Scientists estimate that deep vertical migration transports approximately 15 to 40% of the organic carbon that sinks from the surface ocean to depth each day, a massive movement of carbon that helps regulate atmospheric carbon dioxide levels over long timescales. The daily migration of billions of small creatures has global implications for climate and the carbon cycle. Within the mesopelagic zone where many of these migrating organisms spend their days, we find one of the ocean's most fascinating and challenging environments to study. This twilight zone is defined by its dim, ever-decreasing light. Enough light penetrates to this depth that organisms can still see, but barely. This creates intense evolutionary pressure related to light, vision, and camouflage. Many mesopelagic fish have developed extraordinary eyes, some with tubular eyes that point upward, maximized to collect as much downwelling light as possible to spot the silhouettes of prey or predators above. These tubular eyes cannot move in their sockets, but they're packed with light-sensitive cells at densities far exceeding those in human eyes, allowing the fish to see in light levels equivalent to starlight or dimmer. But having excellent vision is only useful if there's something to see, and many mesopelagic organisms have become masters of invisibility. One of the most remarkable adaptations is called counterillumination. Many fish, squid, and shrimp in the twilight zone have light-producing organs called photophores on their undersides. These photophores emit a glow that precisely matches the color and intensity of the faint downwelling light from above. When a predator looks up from below, instead of seeing a dark silhouette against the lighter surface water, which would make the animal stand out clearly, it sees nothing, because the animal's underside produces light that makes it blend perfectly with the background. It's active camouflage, a biological version of a stealth technology, and it's so effective that the animal essentially disappears when viewed from below. The photophores can be adjusted in brightness to match changing light levels as the animal rises or descends, maintaining the camouflage throughout the water column. Another adaptation to the twilight zone involves transparency. Many organisms in these depths, jellyfish, certain shrimp, larval fish, and others, have evolved bodies that are nearly completely transparent, like living glass. Their tissues are almost entirely water with very few pigmented cells, allowing light to pass through them without creating a shadow or silhouette. Some transparent organisms have only a few visible structures, perhaps just the eyes and a small digestive tract, the rest of the body essentially invisible. This transparency works as camouflage in the dim light of the mesopelagic zone but becomes less effective in the pitch darkness of the deeper ocean or in the bright light of the surface, which is one reason these transparent organisms tend to remain within a specific depth range where their particular adaptation provides the greatest advantage. The twilight zone also hosts a remarkable diversity of gelatinous organisms, jellyfish, comb jellies, salps, and siphonophores, that have adopted a body plan perfectly suited to the deep ocean's low-food environment. Being gelatinous, composed mostly of water with minimal protein or structural tissue, these organisms are extremely energy efficient to maintain. They can grow to large sizes while expending very little energy on body maintenance. Some siphonophores, colonial organisms made up of many individual specialized polyps working together, grow to extraordinary lengths. The longest siphonophore ever documented was discovered in 2020 off the coast of Australia, forming a spiral structure estimated to be about 45 to 50 meters long, longer than a blue whale, potentially one of the longest animals ever recorded. These enormous gelatinous creatures drift through the twilight zone, their stinging tentacles trailing to capture small prey, their translucent bodies nearly invisible in the dim water. And so we've discovered the ocean's greatest migration, billions of tons of life rising and falling daily through hundreds of meters of water, following the ancient rhythm of day and night, creating phantom layers on sonar screens, pumping carbon from surface to depth. We've learned of the twilight zone where these migrants spend their days, where eyes grow enormous and tubular to catch fading light, where creatures produce counter-illumination to erase their shadows, where bodies become transparent as glass, where gelatinous giants drift longer than whales, all adaptations to this realm of perpetual twilight, this boundary between the lit world and the eternal darkness below. Feel your own eyes growing heavy now, your own vision dimming as sleep approaches, like descending from daylight into twilight into darkness. Let yourself drift like a transparent jellyfish, weightless and peaceful, feeling nothing but the gentle rhythm of your breath, rising and falling like the ocean's own vertical migration, natural as tides, ancient as life itself. As we descend beyond the twilight zone, we enter the bathypelagic zone, the midnight zone, where the last traces of sunlight finally disappear completely. This realm, stretching from approximately 1,000 meters down to 4,000 meters, exists in absolute, permanent darkness. No glimmer of surface light ever penetrates to these depths, no daily cycle of brightening and dimming, no seasons of longer or shorter days. It is an eternal midnight, unchanging for millions of years. In this total darkness, the rules of survival change dramatically. Vision, while still present in many species, works only for detecting bioluminescence, the living light produced by other organisms. Eyes in the midnight zone tend toward extremes, either highly developed to detect the faintest bioluminescent flashes, or reduced to nearly nothing, abandoned as unnecessary in an environment where there's nothing to see. Some fish in the bathypelagic zone have evolved what scientists call super black skin, tissue that absorbs more than 99.5% of light that strikes it, making them virtually invisible even when other organisms' bioluminescence illuminates them. This ultra-black coloration comes from a specialized arrangement of melanin particles in the skin that trap light through multiple reflections, preventing it from bouncing back out. Even under bright lights in a laboratory, these fish appear as flat black silhouettes, their three-dimensional features impossible to discern, as if they've absorbed not just light but depth itself. The challenges of finding food in the midnight zone have driven the evolution of some of the ocean's most bizarre feeding adaptations. Perhaps the most famous is the anglerfish, particularly the deep-sea species where the female carries a bioluminescent lure, a modified dorsal fin spine tipped with a glowing bulb that dangles in front of her mouth like a fishing rod. The light is produced by symbiotic bacteria living in the lure, bacteria that the anglerfish cultivates and nourishes. Small fish or crustaceans, seeing this light in the darkness and perhaps mistaking it for prey or a potential mate of their own species, swim closer to investigate, and are suddenly engulfed by the anglerfish's enormous mouth, which can open wide enough to swallow prey nearly as large as the anglerfish itself. The anglerfish doesn't chase prey in this food-poor environment. Instead, it waits with infinite patience, barely moving, conserving energy, letting the lure do the work of attracting meals. But the anglerfish story becomes even stranger when we consider reproduction. The deep ocean is so vast and food is so scarce that males and females of the same species might live their entire lives without encountering each other, making reproduction seemingly impossible. Deep-sea anglerfish have evolved an extraordinary solution to this problem: extreme sexual dimorphism and parasitic mating. The females grow to be relatively large, sometimes 30 centimeters or more, but the males remain tiny, often just a few centimeters long. When a male encounters a female, which may be the only time in his life he finds one, he bites onto her body and doesn't let go. Over time, his tissues fuse with hers, their circulatory systems merge, and he becomes a permanent parasite, or perhaps more accurately, a permanent appendage. The male loses most of his internal organs, degenerating until he's little more than a pair of gonads attached to the female's body, receiving nutrients from her bloodstream and providing sperm whenever she's ready to reproduce. A single female may carry multiple males fused to her body, ensuring she always has access to sperm in an environment where finding a mate twice would be nearly impossible. The midnight zone is also home to some of the ocean's most efficient predators, including various species of deep-sea squid. These squid have evolved hunting strategies that take advantage of the darkness and the sparse distribution of prey. Some species have bioluminescent displays that they can flash rapidly, potentially stunning or confusing prey. Others have enormous eyes. The giant squid and colossal squid have the largest eyes in the animal kingdom, reaching diameters of 25 to 30 centimeters, about the size of a dinner plate. These enormous eyes aren't just for show. They're highly sensitive instruments that can detect the faint bioluminescent glow of prey from great distances in the darkness. Research using optical models of these eyes suggested that a giant squid could potentially detect the silhouette of a sperm whale, one of its primary predators, from up to 120 meters away based on the bioluminescent disturbance the whale creates as it swims through waters containing light-producing plankton. Deep-sea squid and their relatives, the octopuses, have another remarkable adaptation. They've evolved metabolic strategies that allow them to survive in the low-oxygen waters of the deep ocean. Many parts of the deep ocean have lower oxygen concentrations than surface waters because the oxygen dissolved at the surface gets consumed by organisms as the water mass ages and moves through the deep ocean circulation, and there's no photosynthesis at depth to replenish it. Some deep-sea cephalopods have evolved specialized blood chemistry. Their blood contains hemocyanin instead of hemoglobin, and the deep-sea versions of this copper-based oxygen-carrying molecule are particularly efficient at extracting and holding onto oxygen even when environmental oxygen is scarce. They've also evolved low metabolic rates, living life in slow motion compared to their shallow water relatives, conserving energy and oxygen in an environment where both are precious. As we descend deeper still, passing through the bathypelagic zone toward the abyssopelagic zone, from 4,000 to 6,000 meters, we find an environment that pushes the boundaries of what seems physically possible for complex life. The pressure at these depths reaches 400 to 600 atmospheres, the temperature hovers just above freezing, food becomes even more scarce, and yet organisms persist. Among the most successful inhabitants of these extreme depths are the amphipods, small crustaceans, relatives of the beach-dwelling sandhoppers but adapted to crushing pressure. Some amphipod species have been found at depths exceeding 10,000 meters in ocean trenches. What's remarkable is how they've adapted to pressure not through rigid armor, which would be crushed, but through flexibility and special biochemical adaptations. Their cells contain high concentrations of a molecule called piezolite, particularly trimethylamine N-oxide, or TMAO, which counteracts the pressure's effects on proteins and membranes, allowing normal cellular function to continue even under extreme pressure. The deeper the amphipod lives, the higher its TMAO concentration, a direct adaptation to its depth zone. The discovery of these pressure-adapted organisms led researchers to ask a fundamental question: is there a depth limit to life? Is there a point where the pressure becomes too great, the food too scarce, the conditions too extreme for any organism to survive? Expeditions to the deepest ocean trenches sought to answer this question. The Hadopelagic Zone, named after Hades, the Greek underworld, includes the ocean's deepest trenches, from 6,000 meters down to the deepest point at nearly 11,000 meters. Researchers using specialized landers and deep-diving vehicles repeatedly found life even at these extreme depths. Amphipods, sea cucumbers, and specially adapted snailfish have all been documented living below 8,000 meters, with snailfish filmed at depths exceeding 8,100 meters in the Mariana Trench. These Hadal Zone organisms represent the current known depth limit for fish, though simpler organisms like amphipods have been found even deeper. The snailfish at these depths are gelatinous and nearly transparent, with poorly calcified skeletons and bodies more than 70% water, adaptations that minimize the energetic cost of maintaining tissues under extreme pressure. And so we've descended through the midnight zone where darkness reigns eternal, where super black fish absorb all light, where anglerfish dangle glowing lures in infinite patience and males fuse permanently to females in the vast emptiness. We've learned of giant squid with eyes the size of dinner plates, seeing the faint glow of whales from over 100 meters away, of blood evolved to extract oxygen from water that is almost none, of amphipods with special molecules protecting their cells from crushing pressure. We've descended toward the trenches themselves, the Hadal Zone named for the underworld, where gelatinous snailfish swim in near-total darkness at depths that would instantly destroy most life, yet they persist, they thrive, They represent the deepest limit of complex life on Earth. Feel the pressure of sleep upon you now, not crushing but comforting, like the deep ocean pressure that simply is, that organisms adapt to, that becomes normal when it's all you've ever known. Let yourself sink to your own deepest zone, where consciousness fades and dreams begin, where the pressure of wakefulness finally releases and sleep, dark and complete, embraces you. As we continue our exploration of the deepest ocean, we turn our attention to the ocean trenches themselves, those dramatic scars in the seafloor where tectonic plates meet and one plate slides beneath another in a process called subduction. These trenches are the deepest features on Earth's surface, narrow, steep-sided valleys that plunge to depths that dwarf even the tallest mountains. The deepest of all is the Mariana Trench in the western Pacific, where the Pacific Plate subducts beneath the smaller Mariana Plate, creating a chasm that reaches down nearly 11,000 meters at its deepest point, the Challenger Deep. To put this depth in perspective, if you placed Mount Everest, the tallest mountain on Earth at 8,849 meters, at the bottom of the Challenger Deep, its peak would still be covered by more than 2,000 meters of water. The trench itself is more than 2,500 kilometers long but only about 70 kilometers wide on average, creating a narrow ribbon of extreme depth. The walls of the trench slope down at steep angles, sometimes approaching vertical in places. As you descend the trench walls, you pass through every ocean zone we've discussed, from the sunlit surface through the twilight mesopelagic, into the midnight bathypelagic, through the abyssopelagic, and finally into the hadal zone unique to these trenches. Each zone brings increasing pressure, decreasing temperature, diminishing food, and yet the journey reveals that life persists at every depth. The trenches are not just the deepest parts of the ocean, they're also among the most geologically active. The process of subduction that creates trenches also generates earthquakes, some of the most powerful on Earth. The 2011 Tohoku earthquake off Japan, which triggered a devastating tsunami in the Fukushima nuclear disaster, occurred where the Pacific Plate subducts beneath the North American Plate at the Japan Trench. The earthquake released energy equivalent to approximately 600 million times the energy of the atomic bomb dropped on Hiroshima, rupturing a fault zone 500 kilometers long and 200 kilometers wide. The seafloor moved as much as 50 meters horizontally and up to 8 meters vertically in a matter of minutes. For the deep-sea organisms living in and around the trench, such earthquakes are part of their environment, catastrophic events that periodically reshape the seafloor, trigger underwater landslides, and create turbidity currents that rush down the trench walls carrying sediment and organic matter. These turbidity currents, underwater avalanches of sediment suspended in water, play a crucial role in delivering food to trench ecosystems. While marine snow provides a baseline level of nutrition drifting down from above, turbidity currents can deliver massive pulses of organic matter, including plant material from land, wood, and terrestrial organisms that have been carried to the ocean and swept into the trench. Analysis of sediments from deep trenches revealed surprisingly high amounts of terrestrial organic carbon, suggesting that trenches receive more land-derived material than the surrounding abyssal plains. This terrestrial material provides an additional food source that may help support the surprising abundance of life found in trenches despite their extreme depth. Among the most remarkable inhabitants of the trenches are the hadal snailfish we mentioned earlier, but their biology deserves deeper exploration. research on these snailfish revealed that they possess several extraordinary adaptations to extreme pressure. Their bones are poorly mineralized, almost cartilaginous, which makes them flexible rather than rigid. Rigid structures would be more vulnerable to pressure-induced damage. Their bodies contain extremely high levels of TMAO, the pressure protective molecule we discussed, at concentrations far higher than any shallow water fish. But perhaps most remarkably, research published in 2017 identified specific genetic adaptations in the snailfish genome related to pressure tolerance. The genes that code for proteins involved in cell membrane structure, cellular scaffolding, and osmotic balance have evolved in ways that make these proteins function properly under extreme pressure where their shallow water counterparts would fail. These fish aren't just tolerating extreme pressure, they're specifically adapted to it at the genetic level, with modifications that have accumulated over millions of years of evolution in the depths. But there appears to be a physiological limit to how deep fish can live, and researchers believe they've identified why. The limit seems to be around 8,000 to 8,500 meters, which corresponds to the depth where TMAO can no longer effectively protect cellular proteins from pressure damage, no matter how concentrated it becomes. At the concentrations needed to protect against even greater pressures, TMAO itself begins to interfere with normal cellular function. The protective molecule becomes part of the problem. Additionally, at extreme depths, the pressure begins to affect the structure of proteins in ways that no known biological mechanism can fully counteract. This suggests that around 8,000 to 8,500 meters represents a fundamental biochemical barrier for fish and possibly other complex vertebrates, those simpler organisms like amphipods and sea cucumbers, with their less complex physiology, can survive deeper. Beyond the biological limits, the deepest parts of ocean trenches also reveal something about Earth's geological processes and even its environmental contamination. Sediment cores taken from the Mariana Trench and other deep trenches contain layers that record major geological events, volcanic eruptions, earthquakes, climate changes, stretching back millions of years. The trenches act as sediment traps, accumulating material that can be read like pages in Earth's history book. But disturbingly, analysis of these deep sediment cores and of organisms living in trenches has revealed the presence of persistent organic pollutants, chemicals like PCBs and flame retardants that were manufactured on land decades ago, banned in many countries, but which have made their way into the ocean and eventually settled into the deepest trenches. Amphipods collected from the Mariana Trench were found to have PCB concentrations in their tissues comparable to or exceeding those found in amphipods from some of the most polluted coastal areas on Earth, demonstrating that even the most remote and inaccessible places on the planet are not immune to human-caused contamination. The study of ocean trenches has also revealed their importance as sites of carbon burial. The sediments accumulating in trenches contain high concentrations of organic carbon, both from marine sources and from terrestrial material swept in by currents. This carbon, buried under ever-increasing layers of sediment, becomes isolated from the atmosphere for millions of years or more. Over geological time, some of this buried carbon is carried deep into Earth's mantle through the subduction process, effectively removing it from the surface carbon cycle. Researchers estimate that ocean trenches account for a significant fraction of the organic carbon burial in the ocean, despite representing less than 2% of the ocean's total area. This makes trenches important not just as extreme habitats for specialized organisms, but as components of Earth's long-term carbon cycle and climate regulation. And so we've reached the trenches, those narrow scars deeper than mountains are tall, where tectonic plates collide and earthquakes release unimaginable energy, where turbidity currents rush down slopes carrying food from distant lands. We've learned of snailfish with flexible bones and pressure-adapted genes swimming at the edge of what vertebrate physiology can withstand, of biochemical limits that even evolution cannot surpass, of pristine depths contaminated by human chemicals from decades past, and of carbon burial that shapes climate over millions of years. The trenches are at once the most extreme and the most revealing places in the ocean, showing us both the limits and the resilience of life. Feel yourself settling into the deepest trench of sleep now, that place of absolute rest where pressure becomes comfort and darkness becomes peace. Your body knows these depths, knows how to let go completely, knows that the deepest rest is the most restorative. Sink down now, down to where even thoughts dissolve, down to the quiet bottom where sleep is total and complete. As we approach the completion of our journey through the deep ocean, we turn to one of the most exciting frontiers in ocean science, the discovery that the deep ocean seafloor harbors not just isolated vents and seeps, but entire underwater landscapes we're only beginning to understand. Among the most intriguing of these are the deep-sea coral reefs, ecosystems that challenge our very definition of what a coral reef can be. When most people think of coral reefs, they imagine the warm, shallow, sunlit waters of tropical seas, places like the Great Barrier Reef where colorful corals grow in profusion and fish dart among the branches. But in the 1990s and early 2000s, as deep-sea exploration expanded, researchers discovered extensive coral formations growing in the cold, dark waters of the deep ocean, at depths ranging from 200 to over 2,000 meters. These deep-sea corals, unlike their shallow water cousins, don't rely on symbiotic algae for nutrition. There's no sunlight for photosynthesis at these depths. Instead, they're entirely dependent on capturing food from the water, plankton, marine snow, and organic particles that drift past in the slow deep ocean currents. Some deep-sea coral species grow as individual colonies on rocky outcrops, while others form extensive reef structures that can stretch for kilometers, creating complex three-dimensional habitats that support diverse communities of fish, crustaceans, and other organisms. What makes these deep-sea coral reefs particularly remarkable is their age and growth rate. While shallow water corals can grow several centimeters per year, deep-sea corals grow extraordinarily slowly, some species adding only a few millimeters of growth per year, or even less. Radiocarbon dating of deep-sea coral skeletons has revealed individual colonies that are hundreds or even thousands of years old. A black coral collected off Hawaii was dated at over 4,000 years old, making it one of the oldest continuously living organisms on Earth. Some deep-sea coral reefs may have been growing continuously for tens of thousands of years, predating human civilization, their slow accumulation of calcium carbonate creating structures that rose centimeter by centimeter across spans of human history we can barely comprehend. The discovery of these ancient deep-sea coral ecosystems came with a sobering realization: they're being destroyed faster than they're being discovered. Deep-sea bottom trawling, a fishing method where heavy nets are dragged across the seafloor to catch bottom-dwelling fish, destroys coral structures that took millennia to grow in a matter of minutes. Studies using submersibles to survey areas before and after trawling showed complete devastation of coral formations, the ancient structures reduced to rubble. Because deep-sea corals grow so slowly, recovery is measured not in years or decades but in centuries or millennia. A coral reef damaged today may not recover within the lifetime of 100 future generations. This led to increasing calls for marine protected areas in the deep ocean, places where trawling and other destructive activities would be prohibited, though implementation and enforcement in international waters remains challenging. Beyond coral reefs, researchers have also discovered unexpected features on the deep ocean floor that reveal the dynamism of this supposedly static environment. Underwater volcanoes, called seamounts, rise from the abyssal plains, some reaching to within a few hundred meters of the surface while others remain deep beneath the waves. Scientists estimate there may be 100,000 or more seamounts in the world's oceans, though only a small fraction have been studied. These underwater mountains create local environments quite different from the surrounding seafloor. Currents flowing around the seamount bring nutrients up from deep water, creating areas of enhanced productivity that attract fish, marine mammals, and seabirds. Many seamounts are hotspots of biodiversity, with high numbers of endemic species, organisms found nowhere else on Earth. A single seamount might harbor dozens of species unique to that particular underwater mountain, isolated populations that have evolved in place for millions of years. The study of seamounts has also revealed something surprising about deep-sea speciation, the formation of new species. In the deep ocean's seemingly uniform environment, seamounts act as islands of habitats separated by expanses of abyssal plain that many seamount-adapted species cannot or do not cross. This isolation, combined with the long lifespans and slow reproduction rates of many deep-sea organisms, creates conditions where populations on different seamounts can diverge genetically over time, eventually becoming distinct species. Genetic studies of organisms collected from different seamounts have found patterns consistent with this seamount isolation hypothesis, with populations on nearby seamounts sometimes being more closely related to each other than to populations on distant seamounts, but showing clear genetic differentiation nonetheless. The deep ocean, far from being a uniform plane where organisms mix freely, turns out to be a patchwork of isolated habitats that promote biodiversity through geographic separation. Another remarkable discovery about the deep ocean relates to sound. Sound travels exceptionally well through water, much better than through air, and the deep ocean acts as a massive sound channel. At certain depths, typically around 1,000 meters, there exists what's called the SOFAR channel, sound fixing and ranging channel, where sound waves can travel for thousands of kilometers with minimal loss. This occurs because sound speed in water depends on temperature and pressure, both of which change with depth, and at the depth where these effects balance, sound becomes trapped in a horizontal channel, unable to escape upward or downward, instead propagating horizontally for enormous distances. Researchers have demonstrated that low-frequency sounds generated in the SOFAR channel can be detected literally halfway around the world. This has profound implications for marine life. Whales, particularly large baleen whales like fin whales and blue whales, produce low-frequency calls that can enter the SOFAR channel and potentially allow communication across entire ocean basins. A whale singing off the coast of California might, in theory, be heard by another whale near Japan, a communication range measured in thousands of kilometers. This global sound channel also means that the ocean carries acoustic signatures of human activity to enormous distances. Ship traffic, seismic surveys for oil and gas exploration, military sonar, and other human-generated sounds propagate through the SOFAR channel and increase the background noise level throughout the ocean. Studies comparing underwater sound recordings from the 1960s to the present day show that ocean noise levels have increased significantly, particularly at the low frequencies that many marine mammals use for communication. This rising noise pollution may interfere with whale communication, making it harder for individuals to find each other across the vast ocean, potentially affecting mating success and population connectivity. Some researchers have suggested that the global decline in certain whale populations, even in areas where hunting has ceased, might be partly due to acoustic interference, making it harder for scattered survivors to locate each other in the increasingly noisy ocean. The deep ocean also serves as an archive of climate history through another medium, manganese nodules. These are potato-sized lumps of metal-rich minerals that form slowly on the abyssal plains, accreting layer by layer around a small nucleus, perhaps a shark tooth or a fragment of shell. The nodules grow at extraordinarily slow rates, typically just a few millimeters per million years, making them among the slowest growing geological formations on Earth. Because they grow so slowly and continuously, the layers within a nodule record millions of years of ocean chemistry. Scientists can cut a nodule in half and read its concentric rings like a tree, each layer recording the chemical conditions of the ocean when it formed. These nodules have revealed information about past ocean temperatures, oxygen levels, and the cycling of metals through the ocean over timescales of tens of millions of years. But manganese nodules have also attracted commercial interest because they contain valuable metals including nickel, copper, cobalt, and rare earth elements. Mining companies have proposed harvesting nodules from the abyssal plains, but the ecological consequences of such mining remain poorly understood and highly controversial. And so we've discovered landscapes in the deep that mirror those on land, coral reefs thousands of years old growing millimeter by millimeter in the darkness, seamounts rising like mountains creating islands of life in the abyss, sound channels carrying whale songs across entire oceans, and nodules growing so slowly they measure time in millions of years, each layer a page in the ocean's long memory. We've learned that the deep ocean is not simple or uniform but complex and varied, not silent but filled with sound traveling distances we can barely imagine, not unchanging but growing, evolving, responding to forces from within and from the surface world above. Let the slow growth of deep-sea coral remind you that rest is never wasted time, that the stillness of sleep is when you build yourself, layer by layer, restoring what the day has depleted. Like nodules forming ring by ring, your body forms itself anew each night in the quiet depths of sleep. Sink down now into that restorative darkness, that place where time moves differently, where you grow and heal in ways too slow to measure but too important to neglect. As we near the end of our journey through the deep ocean, we arrive at some of the most recent and forward-looking discoveries, insights that are reshaping how we think about the ocean's role in Earth's systems and what the future holds for this vast, mysterious realm. One of the most significant recent discoveries relates to the ocean's role as a buffer against climate change. The ocean has absorbed approximately 90% of the excess heat trapped by greenhouse gases since the 1970s, acting as a vast thermal reservoir that has slowed the rate of atmospheric warming. Without the ocean's heat absorption, the increase in global air temperature would have been far more severe. But this service comes at a cost. As the ocean warms, it expands. Warm water takes up more volume than cold water, and this thermal expansion accounts for roughly 1/3 to 1/2 of the sea level rise we've observed in recent decades, with melting ice contributing the remainder. The deep ocean, that cold, stable realm we've been exploring, is gradually warming too, though much more slowly than the surface. Measurements taken over decades show that even at depths of 2,000 to 4,000 meters, water temperature has increased by a few hundredths of a degree Celsius, a small change, but significant given the enormous volume of the deep ocean and the energy required to warm it. The ocean has also absorbed approximately 30% of the carbon dioxide that human activities have released into the atmosphere since the Industrial Revolution. This CO2 absorption has helped slow the rate of atmospheric CO2 increase in climate warming, but it creates another problem: ocean acidification. When carbon dioxide dissolves in seawater, it forms carbonic acid, which lowers the ocean's pH. Since the beginning of the industrial era, ocean surface pH has decreased by about 0.1 units, from approximately 8.2 to 8.1. This might sound small, but pH is a logarithmic scale, so this represents approximately a 30% increase in acidity. The deep ocean, connected to the surface through the slow thermohaline circulation we discussed earlier, is also gradually acidifying, though the full effects won't be felt for centuries as surface water mixes downward. Ocean acidification makes it harder for organisms to build shells and skeletons from calcium carbonate. The impacts are already being observed in pteropods, tiny swimming snails that are a crucial food source for many fish, whose shells are dissolving in acidified waters in some regions. These changes to the deep ocean's chemistry and temperature are occurring against the backdrop of another recent discovery. The deep ocean contains far more life than we previously imagined, particularly microbial life. Advanced genetic sampling techniques developed in the 21st century have revealed that the sediments of the abyssal plains and the rocks of the oceanic crust harbor vast communities of bacteria and archaea, single-celled organisms living within the seafloor itself, sometimes buried hundreds of meters deep beneath the sediment. These subsurface microbes live at extraordinarily slow metabolic rates, dividing perhaps once per century or even once per millennium, existing in a state between what we typically consider living and dormant. Estimates suggest that the total biomass of these subsurface microbes in marine sediments might rival or even exceed all the microbial life in the entire volume of seawater above. The deep biosphere, as it's called, represents a massive reservoir of life hidden within the planet itself, mostly beyond the reach of photosynthesis, living on chemical energy and organic matter buried in sediments millions of years ago. The discovery of this deep biosphere has led to fascinating questions about the limits and distribution of life on Earth. Cores drilled from the ocean floor have found living microbes at depths of more than 2,500 meters beneath the seafloor, over 1.5 miles down into the earth beneath the ocean floor itself, where temperatures approach 120 degrees Celsius, the upper temperature limit for known life. These microbes survive in tiny pore spaces between rock grains, in cracks and fractures in the oceanic crust, living in total isolation from the surface world, their energy coming from chemical reactions between rock and water. Some researchers estimate that there could be as much as 2 billion cubic kilometers of habitable space within the oceanic crust, a vast hidden frontier that we've barely begun to explore. The deep biosphere might represent more than half of all the microbial cells on Earth, a shadow ecosystem beneath our feet, beneath the ocean, living according to rules and timescales utterly different from the surface world. As we look to the future of deep ocean exploration, technology continues to evolve. Autonomous underwater vehicles are becoming more sophisticated, capable of longer missions and more complex decision-making. Some AUVs can now explore the deep ocean for months at a time, making their own navigation decisions, identifying interesting features to investigate, collecting samples, and mapping the seafloor in detail that was impossible just decades ago. Machine learning and artificial intelligence are being incorporated into deep-sea research, helping to analyze the vast amounts of video and sonar data collected during expeditions, identifying organisms, classifying habitats, and finding patterns that human researchers might miss. There's even discussion of establishing permanent deep-sea observatories, research stations on the seafloor equipped with cameras, sensors, and instruments that could monitor the deep ocean continuously for years or decades, watching the slow rhythms of deep-sea life and detecting changes as they occur. One of the most ambitious visions for deep ocean science involves not just exploring but potentially utilizing deep ocean resources in sustainable ways. Beyond the controversial ideas of mining manganese nodules or harvesting methane hydrates, some researchers are investigating whether the deep ocean could serve humanity in other ways. The cold, stable temperatures of the deep ocean have inspired proposals for deep-sea data storage facilities, where the consistent environment could help preserve digital information for centuries. The unique organisms of hydrothermal vents and cold seeps have already provided valuable biochemicals. Enzymes that function at high temperatures or in extreme conditions have applications in medical research, industrial processes, and biotechnology. Some antibiotic compounds and anti-cancer drugs have been derived from deep-sea organisms. The deep ocean, in this view, represents not just a wilderness to be protected but a library of biological innovations waiting to be studied and potentially carefully utilized. Yet alongside these technological ambitions comes a growing recognition of how little we truly know and how important it is to protect the deep ocean even as we explore it. Recent estimates suggest that we've explored less than 5% of the ocean floor in any detail, and less than 1% intensively. More people have walked on the moon than have descended to the Challenger Deep. We've likely discovered only a fraction of the species that live in the deep ocean. Some researchers estimate that we've cataloged perhaps 10 to 20% of deep-sea species, meaning hundreds of thousands or even millions of species remain unknown to science. Many of these species may have extremely limited ranges, living perhaps only on a single seamount or in a single vent field, making them vulnerable to extinction from localized disturbances before we even know they exist. This incomplete knowledge creates a profound ethical challenge. How do we protect what we don't yet fully understand? The deep ocean faces increasing pressures from climate change, ocean acidification, plastic pollution, noise pollution, and potential deep-sea mining, yet we lack comprehensive data on how deep-sea ecosystems will respond to these stresses. International efforts are underway to establish better governance and protection for the deep ocean, particularly in areas beyond national jurisdiction, the high seas and the deep seabed, which represent the largest wilderness on Earth yet have minimal legal protection. The United Nations has been negotiating a treaty on biodiversity beyond national jurisdiction, aiming to create a framework for establishing marine protected areas in the deep ocean and regulating activities that might harm these ecosystems. The outcome of these negotiations may determine whether future generations inherit a deep ocean still rich with ancient corals and undiscovered species, or an ocean diminished by exploitation and neglect. Voice becomes even softer, slower, more gentle. And now, as we prepare to complete our journey, let's gather together all that we've learned, all the wonders we've discovered in the deep. We began 100 facts ago on the surface, with the HMS Challenger lowering weighted ropes into unknown depths, discovering an abyss deeper than mountains are tall. We descended through the zones, from the sunlit surface through the twilight mesopelagic where creatures migrate daily, into the midnight bathopelagic where bioluminescence provides the only light, down to the abyssal plains where sediment accumulates millimeter by millimeter across millions of years, and finally to the trenches, those deepest scars where life persists at the very edge of possibility. We discovered ecosystems powered not by the sun but by Earth's internal heat and chemistry, black smokers and tube worms, cold seeps and ice worms, evidence that life's possibilities extend far beyond what the surface world suggests. We learned of pressure adaptations and superblack skin, of giant squid with eyes the size of dinner plates, of anglerfish with their living lures and their parasitic mates. We found ancient coral reefs growing in darkness, seamounts creating islands of biodiversity in the abyss, and sound channels carrying whale songs across entire oceans. We discovered that the deep ocean is not separate from us but intimately connected, absorbing our heat and carbon dioxide, collecting our plastic waste in the deepest trenches, responding slowly but inexorably to the changes we impose from above. We learned of microbes living within the rocks themselves, dividing once per 1,000 years, representing perhaps half of all the microbial cells on Earth, a shadow biosphere beneath the ocean floor. And we learned that despite more than a century of exploration since the Challenger, we've barely begun to understand the deep ocean, that thousands or millions of species remain undiscovered, that vast regions have never been seen by human eyes, that the largest habitat on Earth remains the least known, the least protected, the most mysterious frontier not on another planet but right here beneath the waves. 100 facts, woven together into a single story, the story of the deep ocean, the story of life's resilience and adaptability, the story of a wilderness that covers most of our planet yet remains largely unexplored. A story of darkness and cold and crushing pressure transformed into opportunity and habitat. A story of slow growth and ancient rhythms, of connections spanning ocean basins and geological timescales. But most importantly, it's a story that continues. The deep ocean has been here for billions of years and will be here billions more, slowly circulating, slowly changing, carrying within it the memory of Earth's past and the seeds of its future. Whether we choose to protect this realm or exploit it, to study it with humility or disturb it with carelessness, will be part of our own story, our own legacy. For now, though, let these thoughts drift away like marine snow sinking slowly through the water column. Let the deep ocean's peace become your peace, its darkness become your darkness, its timelessness become your rest. You've descended far enough. You've learned enough. Now simply be, suspended in the quiet depths of sleep, where time moves slowly, where pressure becomes comfort, where darkness is complete and welcoming. Feel yourself floating now, neutrally buoyant, neither rising nor sinking, perfectly balanced in the midnight zone of sleep. Your breathing is as slow as deep ocean currents, your heartbeat as steady as the ancient tides. All is still. All is dark. All is exactly as it should be. The ocean will be there tomorrow, vast and mysterious as ever, waiting with its untold stories and undiscovered wonders. But tonight, you are deep, you are safe, you are at rest in the abyss. Sleep now, dear listener. Let the gentle pressure of sleep embrace you like water at depth, total, complete, inescapable in the most comforting way. Let your last conscious thoughts dissolve like salt in seawater, becoming indistinguishable from the whole, becoming part of something larger and older and more peaceful than individual consciousness can hold. Thank you for descending with me through these 100 facts about the deep ocean. Thank you for exploring the abyss, for learning about this remarkable frontier, for staying with me through the twilight zone and the midnight zone and all the way down to the trenches. May your sleep be as deep as the Challenger Deep, as peaceful as the abyssal plains, as restorative as the slow circulation that renews the ocean from within. Sweet dreams, dear listener. May you drift gently in currents of rest, floating in darkness that comforts rather than frightens, suspended in the infinite peace of the deep. Good night,